The overall goal of this molecular marker method is to characterize, quantify, and isotopically analyze the pyrogenic carbon fraction in environmental samples.
This method can help answer key questions in various fields, ranging from archaeology and environmental forensics to biochar and carbon cycling research. The main advantages of this technique are detection of pyrogenic carbon over a broad range of the combustion continuum, simultaneous characterization, quantification, and isotopic analysis of pyrogenic carbon, and applicability to a very wide range of environmental sample materials. Demonstrating the sample preparation and chromatography will be Ulrich Hanke, a grad student from our laboratory. The subsequent isotopic analysis of the purified target compounds will be shown by Negar Hagipur, lab coordinator at ETH Zurich.
To begin this procedure, weigh previously freeze-dried and homogenized samples into quartz digestion tubes and cover against dust with aluminum foil.
Use only clean, decalcified and combusted glassware, thoroughly clean tools and ultra-pure chromatography-grade water and solvents for the entire procedure.
Add 2 milliliters of 65% nitric acid into the digestion tubes. Use a vortex mixer to assist thorough wetting of each sample. Following this, insert the digestion tubes into a pressure chamber. Close the pressure chamber according to the manual and place it into a preheated oven at 170 degrees Celsius for 8 hours. After removing the digestion